So I'd like to begin by thanking the organizers for inviting me to this talk. In some ways, I am one of the newest members of this community and also one of the oldest members of this community. I started working on G proteins before they were called G proteins as a graduate student in 1983. But I did not start working on GNAO1 until 40 years later and published our first paper in 2023. I have been working on G proteins for a very long time, and since I'm new to this conference, I thought I would give a talk outlining our approach to understanding G proteins, how they work, and what happens when they're broken. I started off my career working on G protein coupled receptors, and as a graduate student, I participated in the sequencing of the first neurotransmitter binding G protein coupled receptor. I then got interested in switching to try to understand how G proteins work. And one approach is to, if you want to understand a machine, if it's a car, you start taking parts out and you see what's broken. If you didn't know anything about a car, one way to learn about what the parts do is to break them and see how that affects the function of the machine. And so my approach for the last 35 years has been to use genetics to try to break G proteins to understand how they function normally. And for most of that time, it was working with G proteins in a model organism, Baker's yeast Saccharomyces cerevisiae. And then, of course, I became aware of the tremendous genetic diversity, particularly affecting GO, and got interested in trying to apply some of the knowledge that we had gained doing yeast genetics to try to understand human genetics. And as it turns out, many of the proteins that we had studied in yeast, purely because we were interested in how G proteins function, many of those mutations that we have been studying for many years are appearing in these patient databases. And there may be some individuals in this room today that have mutations that we have been studying for decades. So I'm going to give you a little bit of an outline of the type of approach that we're taking. You all know that G protein coupled receptors are tremendously diverse. When I was a grad student, there were just a handful. We now know that there are about 800, and they are very important in pharmacology. They're a third of all drugs, 2/3 of hormones and neurotransmitters. And I think it's important to emphasize that the stimuli that target these proteins are incredibly diverse. Light in our retinas, they have no mass, to steroids, phospholipids, proteins, and so on. You've seen some variation of this slide before, but I'm going to be coming back to it, so I'll just give you an introduction to how I will present it. The green box is the receptor, the neurotransmitter receptor. This is the G protein alpha subunit, GNAO1, beta and gamma subunits, and what the receptor does is stimulate the release of GDP and the binding of GTP. Okay, so it is these dissociated subunits that activate proteins inside the cell. Now, every on-step has an off-step. And if signaling happened all the time, that would be bad. If it never happened, that would be bad. So the timing is very, very important. And one of the things that came out of our work with yeast is the discovery of this RGS protein, which functions as a GTPase accelerating protein. So it speeds up the inactivation of the active G protein back to the inactive state and leading to the reassociation of the subunits. All right, so in the last couple of years, we've become interested in how mutations in genetic mutations in humans can cause disease. And we've done some work with GNAO1. We've also done some work with a cancer-causing mutation in GQ. And I bring this up because I understand that this audience is very interested in GNAO1, but there is a lot to be learned from studying other genetic diseases linked to G proteins and using other genetic model organisms to understand how G proteins work and how they can fail. So perhaps the most well-studied example of a G protein mutation linked to disease is one that affects GQ. And this one was striking because it is genetically homogeneous. So almost all of the people who walk around with this mutation, it causes a type of cancer of the eye, they have a mutation at a single amino acid, and it's glutamine, and it's substituted to just one of two possible alternatives. Very homogeneous. Almost everybody that has this type of disease, there are only two substitutions, one G protein, one site. And I think it's important to contrast this with GNAO1 disorder, which is striking because it is so genetically heterogeneous. It affects both catalytic and non-catalytic sites, including this catalytic glutamine. And I got on ClinVar the other day, and I counted up there are 240 unique variants in GNAO1 that are possibly or likely pathogenic, and they occur at 170 sites across the protein. So this type of heterogeneity, it's extraordinary, and it means that this is almost certainly not a single disease, but a spectrum of diseases, and I think this is important, has important implications for treating the disease, but also understanding the molecular causes of the disease. So the cancer-causing mutations, what they do is they block GTP hydrolysis. It's very simple. The G protein gets stuck in this active GTP-bound state and continues to signal and drive cell proliferation, and you get uncontrolled cell growth and cancer. We did publish a paper on this a couple years ago, and we found that even this disease can be very heterogeneous, and that the non-preferred substitutions behave in ways that are completely surprising and unexpected. And I'll touch on that a little bit in my talk. Now, before I do that, I want to introduce you to this Brett method. And you've seen variants of this already in the talks, but since I'll be coming back to it again and again, I just want to remind you. So one way we measure G protein activation is we track the dissociation, the separation of the alpha and beta gamma subunits. And so we have these tags, and when they're close together, it emits light, and when they're far apart, the light goes away. So when we activate with a drug, we watch the light go down, and this is an indicator of the function of the G protein and their ability to associate with one another. All right, so these are the cancer-causing mutations. I mentioned that it's GQ, and almost always it's a QL or QP substitution. So we thought, what's special about QL and QP, and for that matter, what's special about this G protein subtype compared to other G protein subtypes that exist in humans? So we mutated that Q to all of the other possibilities. There are 20 amino acids, and we replaced all of them, and we did this Brett method. So compared to wild type, in every case, there was a permanent separation of the G protein subunits. This did not surprise us. This is a permanently activated G protein, and the subunits are always apart, and they're always signaling. Now, what was surprising was we look at other G protein subtypes. So GS and GI are two other famous examples. Look at GS. Here, they're not constitutively active. In fact, compared to wild type, when you add a drug, a neurotransmitter, you get further activation of the G protein. And with GI, you get a mix. Some are permanently activated, and some are activated with the addition of a drug. So the surprising thing here is that even a mutation that we think of as being always on, in some cases, in some contexts, in some G proteins, you can get further activation with a drug that targets the receptor. And in principle, you should be able to get G protein inactivation with a drug that targets the receptor. I think this is an exciting discovery because there are not many drugs that target G proteins. There are lots of drugs that target G protein coupled receptors. There are a lot of drugs. They are 1/3 of all FDA-approved drugs. So if you can come up with a drug that indirectly shuts off a mutant G protein, one that causes disease, and targets the receptor, that is a much faster, more direct, and possibly simpler path to a treatment. if you can repurpose drugs that are already FDA approved, already approved by government agencies, then we already know that they're safe. They're okay to give to people. And if we can match FDA-approved drugs with disease-causing mutations in G proteins, that would be a much faster path to treatment. All right, I don't want to spend too much time on this because this is quite technical, but we are biochemists, and we have biophysical methods for measuring the function of the G protein. And another thing that came out of this, and this was done by a method called NMR, each of these dots shows the position of amino acids in this protein, and then we monitor how those amino acids move when you mutate one of the key residues. Okay, and I don't want to spend too much time on this other than to say that people often think that a mutation which puts a G protein in a permanently activated state, that protein looks very much like a normal G protein bound to GTP, and that's a very reasonable assumption, and that was the assumption that I made. But what we learned from these biophysical studies is that that the mutationally activated G protein is in a shape, in a conformation, has different dynamics than the normal wild-type G protein that has been activated by a neurotransmitter. Okay. The lesson here is that even when you have a mutation that is permanently GTP bound, some of these mutations do not activate the G protein, and we also have to consider that because they have different shapes, that they have different functions, they may have different cellular targets. And so there may well be new signaling pathways that don't occur in a normal cell that occur when you mutationally activate a G protein. And I mentioned this before, but G protein mutants, some G protein mutants may be druggable. There are some experimental compounds, zinc, for example, that seem to target the G protein directly. But we know that some of these mutations can be further activated and inactivated with drugs that target the receptors. And there are a lot of drugs that do that. G protein coupled receptors, GPCRs, are far more druggable than G proteins. And I think this is an avenue that we should be exploring. Okay, so I'm going to stop talking about the cancer mutations and focus now on GNAO1 disorder. Genetically heterogeneous. So I'm going to talk very briefly about 3 papers that we have published in the last 2.5 years. This is our foray into GNAO1 disorders. 2 of them, well, all 3 were spearheaded by a talented graduate student, Kevin Knight, who is now in Kirill's lab. And this first one is, I think, quite important because in this paper we took 55 different GNAO1 variants, all linked to disease. We purified the proteins and we studied their function in cells. And not surprising to us, the functional heterogeneity of these G proteins was the thing that popped out. And we attempted to classify them into functionally distinct groups. All right, why is these classifications important? Again, if you are going to do deep brain stimulation, this is an invasive procedure, it has risks. It would be nice to know that other patients that have benefited from deep brain stimulation have a mutation that is classified in the same way that one that affects perhaps a family member that you have. And then our next step is to take some of the best behaving mutants and do a deep dive and try to really figure out structurally, functionally, mechanistically, how these mutations disrupt the function of the G protein. All right, so I'm going to talk about these deep dives. I mentioned that the cancer-causing mutations are concentrated in this step of G protein inactivation. In GNAO1, the mutations affect multiple steps of the G protein activation cycle. There are mutations that are RGS insensitive. There are mutations that are locked in the GTP-bound state. The same mutation that causes melanoma in the eye, in GQ, it appears in ClinVar as a GNAO1 disorder. Oddly enough, it does not cause cancer. It causes GNAO1 disorder. And then this was alluded to in some of the earlier talks. There are some mutations which cause instability. The protein's not expressed. If you don't have enough of the protein, you have too much free beta gamma and uncontrolled signaling downstream. And then I'm going to talk about two other mutations, one that is locked in this complex with receptors, and another that binds GTP but fails to release beta gamma. So with each of these mutations, we can block different steps of the G protein activation cycle and get some clues into how we could perhaps in the future use drugs to bypass these deficits. All right. So I mentioned we studied 55 different variants linked to GNAO1 disorder. And the first group, these are scattered throughout the protein. And we focused on this RAS-like domain because that's the part of the protein that has all of the enzyme function. The group 4 mutants are buried in the protein. They're quite far from the nucleotide binding site, shown here in black. And for the most part, they were very difficult to purify and had no enzyme activity. So these are basically dead proteins. When we do this Brett assay, which I introduced earlier, the wild-type protein, we see a nice release of beta gamma. The signal goes down because the proteins are separated. In every case, these dead proteins, they're completely unresponsive to the drug, and they just lie flat. And this is shown for all of the different variants that we put into group 4. All right, group 3 mutants. This is a group that we have not published on yet, but this is a big emphasis of my lab at the moment. And these mutations are at the surface of the protein and are very likely to be disrupting the interactions with known G protein binding partners, RGS proteins, for example. And then the rest of my talk, I will be presenting on group 1 and group 2 mutants. If you think of this as the layers of the onion, group 2 is close to, but not touching, the nucleotide, the GTP. Group 1 mutants are very close to the GTP and are very likely to be interfering with the ability of this protein to bind and convert GTP to GDP. Okay. So when we look at the function in these cells, the group 2 mutants compared to wild type, shown in black, they can be activated, but the activation is somewhat diminished. They're a little bit less able to respond to the neurotransmitter, and that was shown for all of the group 2 mutants here. The group 1 mutants do something extraordinary. When a receptor activates a G protein, it leads to separation of the subunits. The group 1 mutants, they, upon stimulation of the receptor, lead to association of the G protein subunits. So it's the complete opposite. It's recruiting the G protein to the receptor, and what I'll show you is that it forms a permanent complex with the receptor that cannot be activated. Okay? And I think what's clear from these graphs is when you look at the 4 different groups, they have 4 very different behaviors by this assay. And again, we've also purified these proteins and we've studied their enzyme activity, and the classification scheme holds. Okay, so I'm going to finish by talking about group 1 and group 2. We did a deep dive into a group 1 mutant and a deep dive into a group 2 mutant. So group 1 mutants, our hypothesis is that most or all of them are locked in this receptor-bound complex. When you activate the receptor, this G protein assembles with the receptor, but doesn't let go. It's stuck. Okay? It's not activatable. So you're not doing the normal things that a G protein does. All right. So here's one that we studied. And so this molecule here is GTP. It's a guanine ring, phosphate, phosphate, phosphate. There are 3 phosphates. And proteins don't like negative charges inside the protein. And so G proteins have this lysine. All right? This is a positive charge. You put a positive charge next to a negative charge, and the protein is happy. All right. This Ke substitution takes a positive charge and introduces a negative charge. Negatives don't like to be next to negatives. And so in my mind, there's absolutely no way that this substitution is compatible with GTP binding. And in fact, when we did a deeper dive, we learned that that is in fact the case. All right, I'll get back to that in a moment. Group 2 mutants. So R209C, it's locked in the GTP-bound state, but it doesn't undergo the normal shape changes that are required to release beta-gamma, and so you fail to get activation. Also unprecedented. Here's what it looks like when you study the protein. Here's the GTP. This is the GDP. And the arginine that I'm about to talk to, it's way out here. It's not particularly close, all right? But when you switch GDP for GTP, you see that the arginine and these other amino acids collapse in on the GTP, and this amino acid right here is required for the release of beta-gamma. All right, so you can imagine if you disrupt this bridging molecule and this network, this connection from GTP to the beta-gamma subunits, is broken. And so if you mutate the arginine, you may well have a G protein that binds GTP, and it does, and hydrolyzes GTP to GDP, and it does, but the G protein cannot sense the presence of the GTP and fails to release beta-gamma. All right, so we had an introduction to this approach before. We have a fluorescently labeled GTP that we can measure as it binds to the G protein, and then as it gets converted to GDP, like it's supposed to, that signal decays. This K to E mutant, the group 1 mutant, we thought would not bind GTP, and in fact we see no binding and no hydrolysis. Okay? This is purified protein. The R209C mutant, this is a group 2 mutant. This is the one that we think binds GTP, hydrolyzes GTP, but doesn't let go of beta-gamma. And the black is the wild type, the blue is the mutant. It's a little slower in the hydrolysis, but it is a competent enzyme. This is a perfectly functional enzyme. Okay, the next step is, what about beta-gamma? So again, we're biochemists, we're interested in how these proteins interact, and we always look to have very quantitative measures of enzyme activity. And this is an experiment where we're measuring the ability of the purified alpha and beta-gamma subunits to bind, and we're comparing the wild-type protein with the group 1 mutant and the group 2 mutant. And then what we do is we immobilize beta-gamma on a glass slide, and then we flow different amounts of alpha across the slide, and that changes the diffraction of the light on the slide. And by measuring the binding in response to increasing concentrations of the G protein, we can calculate an affinity of alpha for beta-gamma. And the important thing here is that the affinity for wild-type alpha beta-gamma is no different from the group 1 mutant and no different from the group 2 mutant. So for example, the fact that this cannot bind or hydrolyze GTP, you say, well, it's just a dead enzyme. But it is perfectly competent to bind the beta-gamma subunits. So this protein, it cannot bind GTP, cannot bind GDP, but it can bind beta-gamma just fine. Okay, so the group 1 mutant, one of the ways that pharmacologists nowadays try to find drugs is, in the old days, you would give a rat a whole bunch of chemicals and look for a change, or maybe a cell. But increasingly, pharmacologists are using three-dimensional structures of proteins and computers to dock hypothetical molecules to the protein structure. Now, to do that, you need to solve the structure, and that is a very difficult, time-consuming, expensive process. And with G proteins, it's particularly difficult because the only way you can get the complex of the receptor and the G protein subunits is to add an agonist to the receptor and to remove the nucleotide. That's the form of the complex that is the most stable. So here we have a mutant that doesn't bind nucleotide at all. It binds permanently to the receptor. And I see this as a way to accelerate computer-driven drug discovery by accelerating determinations of three-dimensional structures of G proteins and G protein coupled receptors. So we went down the hall and talked to our neighbor, Brian Roth and Brian Crum, and they took our mutant, shown here in blue, this is the G protein alpha subunit, and combined it with the dopamine receptor. And this is the dopamine, of course, is very important in movement, bound to dopamine, beta and gamma subunits. This is the first three-dimensional structure of a human GO protein. All right? So if you're going to do drug screening computationally or experimentally, you should be working with GO, and here's your structure. It's also the first structure of the D2 dopamine receptor bound to dopamine. Dopamine is very important in movement. And so if we're going to be docking, trying to find drugs that target receptors involved in movement, it's good to have a structure of the native dopamine receptor bound to the neurotransmitter dopamine. Okay? So this is a 4-protein complex. It's the receptor bound to dopamine, the first time we've done that. A lot of these structures, they only show half the G protein. The RAS-like domain, this is one of about 1% of three-dimensional structures that shows the entire G protein. I think this is going to be important. There's a lot of mutations in this all-helical domain. Okay, I think this is supposed to be a movie, but I fear it's probably not going to work. All right, I think it's not going to work. Okay. The last thing I wanted to say about this structure is that we can very nicely see the mutation here, the absence of the guanine nucleotide. We can see dopamine, the drug binding pocket, very nicely in this structure. And then also, for those of you who are interested in G proteins, a very nice opening of the 2 domains of the G protein, allowing the nucleotide to bind into this pocket right here. Okay, so I'm going to summarize my presentation. I told you about mutations in the catalytic glutamine, how they create an ensemble of GTP-activated states. I went into a deep dive of one of the group 2 mutants. This is a mutation that prevents the release of beta-gamma. Too little beta-gamma in the brain, that's a bad thing. It doesn't release beta-gamma even when bound to GTP. And then we did a deep dive into one of the group 1 mutants, lysine 46. This one does not bind GTP at all, does not let go of the receptor. It also ties up all the beta-gamma. This is also bad for the cell. In closing, I just want to emphasize that I think there's a lot to be learned from studying G protein disorders outside of GNAO1 disorder. People are trying to find drugs to treat uveal melanoma, which is caused by GQ, and I think we have a lot of lessons to be learned from talking to the people involved in G proteins and cancers. We're very interested in doing an even more comprehensive profiling of GNAO1 mutants. We've done 60. It's about 1/4 of the total. If I had the resources, I would do all of them because I think this is going to inform treatments in the future. And finally, a drug that works for one mutant is likely not to work for another, and a mutant that doesn't work for one mutant may work for another. So I don't think that we can just think of this as one disease, and just because a drug trial doesn't work for a handful of mutants or works for some of those mutations, that doesn't mean it's not going to work for others. And I really think that the best way to address this issue is to screen as many mutants as possible and look for patterns. And if, for example, deep brain stimulation works for this subset, what do they have in common? And then we can spare patients and their families from treatments that don't work, it's all risk and no reward. I mentioned that group 3 is a big interest of mine. This is what we're working on now. These are the ones at the surface. If anyone is aware of any patients that are affected, have mutations at one of these residues, we're working with Laura and Erica, who are here to try to understand how patient phenotype and molecular mechanism are related. And we are doing some work with G proteins, trying to find drugs that, like RIC8, may be helping the misfolded or mutated G protein restore its function. This is science fiction. This is something that may not help anybody in the future, but I think it's something that, that we can do very well in the lab. And finally, I want to thank members of my lab, shown here. Kevin Knight did spearheaded the GNAO1 work that I talked about. Natalie Hewitt talked about the GQ mutants. These are collaborators that have helped us along the way, some of whom are here. We're very grateful for their collegiality and their partnership. And thank you for your attention.